Okay, good morning, everyone. Thanks for being here. Um, are there any questions from the previous lectures? Good. Well, if not, then I'll get started. Okay. So you're able to see the screen, are you?
Yes.
Okay.
Right.
Good. Today's lecture is on fertilization in cleavage. I'm going to talk about generation of gametes, and so this is a kind of a vague continuation of some of last week's lecture, which touched on the cell cycle and mitosis. I realized I forgot to introduce one important point of mitosis and cell cycle, which was this one. So I just wanted to say that some embryos have an altered cell cycle, and so the early Drosophila embryo, and Tina will probably go into this in much more detail, doesn't have these G1 and G2 phases, okay? And it goes directly from mitosis to the S phase, from M phase to S phase. It does this to generate many, many nuclei. So the early Drosophila embryo doesn't undergo cell division, and the first 13 cell cycles are just going between M and S phase. Okay, and it uses this CDC2 cyclin B mechanism, and cyclin B is degraded to bring mitosis to an end and then go into S phase interphase. Okay, so as well as mitosis, I'm sure all of you have studied meiosis as well. That's correct, yeah? Everyone knows meiosis?
Yes.
Yeah?
Yeah.
Okay, good. Anyone doesn't? Phew. So mitosis is, in the embryo, mitosis generates many, many cells, okay? And this is the mode for somatic cell replication. Meiosis, however, is used for generating gametes, okay? And I think also this is very high school stuff, yeah? So all of you are aware of that. The generation of gametes is interesting, and today we're going to talk about fertilization, but first we should talk a little bit about how gametes are generated. And so just so I can understand what you know, can anyone tell me what feature of meiosis is used particularly for gamete generation?
The genetic material is, if the chromosome number of a cell is 2n, then via meiosis and for gamete formation, it is converted to n.
Yep.
If there is reduction in the genetic material, the chromosome number.
Yeah. So that's one. And another point?
Crossing over also takes place between sister chromatids.
Yeah, fantastic. Okay. So yeah, so definitely, so you're reducing the number of chromosomes from 2n to n, and you're increasing the potential genetic diversity by crossing over during prophase 1. Okay. Yeah. So here, so as well as the kind of reductive cell division, we're also having recombination between sister chromatids during prophase of meiosis 1. And so all of you know I think leptotene, zygotene, pachytene, diplotin, and diakinesis. Is that correct? Is that something everyone is kind of remembering, vaguely familiar about?
Yeah.
Yeah.
Good. Phew. Because I don't really know this very well. Okay, so if you don't know it, contact me later, and we can try and revise it together. Okay, but essentially during this phase of meiosis, so prophase, we're getting recombination events happening between sister chromatids, and these events start early in leptotene. You're getting homologous pairing. You're getting these breaks forming, and these kind of strand inversions, and the very famous holiday junctions being stabilized in late pachytene. Okay, so this is one of the most important features of meiosis, and this is how genetic diversity is brought into gametes. So thinking about this, let's think about how both oocytes and sperm are generated. So the precursor cells for both oocytes and sperm undergo a period of slight mitotic proliferation. Okay. At this point, if you're an oocyte, I'm going to talk about this a little bit more, but sperm will just go straight into meiosis and generate these sperm. Okay, and so you have these spermatogonial stem cells which are undergoing mitosis all the time, and they'll start to undergo meiosis to generate sperm cells. And this is triggered at puberty for us and for mammals in general, and a little bit, and whatever the equivalent is, the metamorphosis stage in other animals. And so this is fairly consistent throughout in all animals. Oocytes are different, and so the oocytes are invested with a lot of so the animal invests a lot of energy into making an oocyte. So this is going to carry forward the next generation. After mitotic proliferation, you begin meiosis. Some of these oocytes get surrounded by follicle cells. These follicle cells are providing nutrients and various other things like restricted mRNAs, yolk, if you're that kind of animal, into the oocyte. And these continue by oogenesis throughout life. At puberty, the oocytes mature, and you'll start getting a mature oocyte which is able to be fertilized. So I want to bring this background into meiosis as well. And so at particular points in the during oogenesis, during the formation of oocytes, meiosis is arrested. And so tell me if any of you know this, or if this is the first time you've heard this.
I did not know this.
Okay.
I heard about in mammals.
Yeah. So in mammals, kind of this meiotic arrest is fairly famous. But not almost every species undergoes a meiotic arrest. Yep. You're good.
Sir, can you hear me?
Yes.
I'm curious, like at the meiosis 2 happens at the puberty in case of females.
Yeah.
So how that cell arrest, I mean, first meiosis stage happens when she was in her mother's womb.
Yeah.
And when she reach at puberty, then second stage of meiosis happens and haploid ovary forms. So how that cell coordination happens here, like cell goes to in arrest stage, and at some point it gets activated and undergoes second meiosis.
Yeah.
I mean, where is the control kind of thing? That's interesting.
So it's easier to talk about the control for meiosis 2. Yeah, this metaphase 2 arrest here, okay, for mammals. And if I told you that this metaphase 2 arrest happens for almost the entire lifespan of the mammal, can you have a guess what the activation for this myosin, this release for meta2 arrest is?
I don't remember at this point.
Okay.
I'm not revised.
So meta1 is released on puberty, okay? This meiotic arrest here, okay?
Mm-hmm.
This arrest here is released on fertilization, okay?
Okay.
Yeah.
Okay.
Yeah. So the coordination for metaphase 1 appears to be kind of course, if it's coming from puberty. Sorry, it's here. It's here. This prophase 1 arrest. This is these are different animals. So it's a prophase 1 arrest, okay, in mammals, and this is happening at puberty. So this is released at puberty, and so it's thought that the luteinizing hormone, which is one of the puberty triggering signals, act on the follicle cells that are around the oocyte, and these signal to the oocyte to kind of relieve, to bring down the levels of cyclic GMP and cyclic AMP. And that's thought to allow this arrested oocyte to then break down its germinal vesicle and mature a little bit more so that it becomes arrested here, okay? Sorry, here in metaphase 2.
Okay.
Is that, yeah?
Yeah, thank you.
Are there any other questions about this point? Good. So what does this mean, okay? What it means is that when we're thinking about an oocyte undergoing meiosis, one oocyte progenitor doesn't give rise to 4 oocytes. It only gives rise to 1 oocyte, okay? And we'll come back to this point in a little while, okay? So what I want to say is that this arrest here, this metaphase 2 arrest here, is released upon fertilization. Upon fertilization, the oocyte completes meiosis and at the same time, simultaneously, fuses with the paternal pronucleus to become a zygote. This primary arrest, this first meiotic arrest, is there for all animals. So all species will arrest at this point. Here you can see which species arrest at different points. So Drosophila, mollusks, and ascidians will arrest here at metaphase 1. We arrest at metaphase 2. Okay.
Raj, what is the full form for GV, BD, and PB1 and 2?
Pardon?
What are the full forms for GV, BD, and PB1 and PB2?
So GVBD is germinal vesicle breakdown. Okay. PB1 is polar body 1, and PB2 is polar body 2. So I'm glad you asked this question, Nivedita, because I answered you incorrectly last time, okay? when you pointed out these two flashes of red outside the C. elegans, and I told you it was a follicle cell. It wasn't. It was a polar body cell. Okay? Yeah, and we'll go back into this in a few slides. Okay. So, like I said, male gametes are not meiotically arrested. They will undergo mitotic proliferation to form spermatogonial stem cells, which upon puberty are then triggered to undergo meiosis. Okay, and these will just go straight into meiosis without any arrest. So, because it's easy to talk about the sperm, I'll just show you this kind of simple representation of what the sperm is. It's basically a vehicle to carry the paternal genome. So the nucleus is highly compacted. A centriole is still attached to the nucleus. You have mitochondria in this shaft here. You have a tail. The head here also consists of the acrosome here, which I'll talk about in the next few slides. So surprisingly, almost all sperm look the same, with the head and a tail. The only animal which has a different morphology for sperm is C. elegans. And it has kind of this amoeboid type sperm, which is shown here. And this kind of crawls on the skin of the roundworm, looking for the oocytes. Okay, but that's the only animal. Yes.
So in general, cell shape is maintained by like cytoskeletal elements, but I think sperm doesn't have that much of cytoskeletal elements. So how is the shape maintained in these cases?
So there is a fair amount of cytoskeleton in here. The tail, of course, consists of microtubules. So it consists of an axoneme, which is very standard. It's conserved between flagella of different bacteria. Chlamydomonas has a flagella, which has the same basic architecture as the sperm tail. The mitochondria here are wrapped around, again, this tail, which is this microtubule axoneme structure. And around here motors called dynein motors, which are powering the movement of the tail. Okay, the head has some microtubules. It has some actin, but you're right, not very much, and it doesn't really need very much. Yeah, the membrane of the sperm is only there to cover it, and everything else is quite tightly packed. Okay. Did that kind of answer your question?
Yeah, go ahead.
Okay, thank you. So this is a sperm. This is a C. elegans sperm. And so let's just basically think about what the issue is here when we're thinking about fertilization. And then I'll come back to the events that happen during fertilization when we're about the oocyte. So this is something I pulled off YouTube. It's a Ascidian embryo, sea biscuit. Here it's laying eggs. You can see this. And here it's releasing sperm. Okay. So before we go any further, maybe we'll go a little bit further. One thing I kind of want to bring up here is that so the Ascidian is lives in is of course a marine animal, and remember these marine animals, they're not solitary. There's many different types of marine animals, all trying to fertilize each other. Sorry, not each other. They're fertilizing. The males are fertilizing the females of their species and things like that. But the sea around there is just a mix of sperm from many different species. And so somehow these sperms manage to find their eggs, and sometimes from far away. And the kind of the chemoattractive signals that are involved, recognition signals that are involved are incredibly fascinating. And that's all I'm going to talk about for the time being. If you're interested, I will put up some references on kind of this sperm egg recognition problem, which is really fascinating. And then this idea of chemoattraction between sperm and egg of the same species, I don't think has been worked on so much. And it's kind of one of the open questions on how you get species specificity during fertilization. So what you're going to see now is fertilization happening. And the sperm has entered, and the first thing you see is the elevation of the vitelline membrane, the vitelline envelope, sorry. And what this is a way of protecting the egg against other sperm entering. So it's a protection against polyspermy. This huge egg, this vitelline membrane is released by cortical granules that are located on the cortex of the oocyte, and the events around fertilization cause these cortical granules just to be emitted, to fuse to the membrane, and then this vitelline envelope kind of solidifies, jellifies around the oocyte. The next thing you'll see is the pronuclei here, maternal, paternal, and then they'll fuse. And once they fuse, you'll start to get cleavage. Okay? So let's talk about these events in a little bit more detail. So this is kind of what you saw. This is a sea urchin. The mammal I'll show you next. As the sperm gets closer to the egg cell membrane, it encounters a jelly layer, which triggers this acrosome to be ejected. Okay, and so here you can see the acrosome. Once the sperm kind of recognizes the jelly layer, this membrane, this acrosome fuses to the membrane and releases what are essentially a cocktail of proteolytic enzymes. At the same time, there's kind of a cap of actin above the nucleus, above the pronucleus. This undergoes some transformation, so it forms a kind of spear, which is called the acrosomal process. And as the enzymes from the acrosome dissolve the jelly layer, the rest of the sperm is powered through the jelly layer with this acrosomal process until it touches the egg cell membrane. Once it fuses with the egg cell membrane, once it touches the egg cell membrane, it fuses, so the membranes fuse, and this causes the release of the paternal pronucleus into the egg. So this is a sea urchin. Mouse is a little bit different. So in mouse, you don't have this acrosomal process. The names are a little bit different, but the process is kind of similar. The egg is surrounded by what's called the zona pellucida, and then this cumulus layer. The sperm can bind the zona pellucida. It's allowed to contact the zona pellucida. Once it does that, you get the same kind of acrosomal reaction, which releases the proteolytic enzymes of the acrosome, allowing then the sperm to basically burrow through the zona pellucida and then touch the egg cell membrane. Once the sperm head touches the egg cell membrane, it fuses and releases the paternal genome, paternal pronucleus into the egg. Do you have any questions at this point? I think this is all fairly easy to understand, but if there's any questions,
so in case of the sea urchin one, yes, slide.
Yeah.
So what triggers the proliferation of the actin to form that protrusion? Like what signal it is getting from the egg?
So I want to say no one knows, but what I really will say is I don't know, okay? And so it's not proliferation of actin, it's more a polymerization of actin. And it could be that the release of the acrosome also changes the internal pH sufficiently to set in motion a particular actin polymerizing event to cause this to happen. Okay. So what we are left with then is
Raj, I had a question.
Yeah, Atal.
Yeah. Like in the cases of humans, we have this prevention of polyspermy happening when the sperm tries to fuse with the zona pellucida. No more sperms can actually fuse with that layer and cross it. So, what modification does the zona pellucida layer undergo such that it does not allow any more sperms to fuse with it?
That's a good question. Let me come to that. Okay.
Okay.
I think that's in the next slide. Okay. So thanks for asking that. Okay. What we have then is the egg that's just being fertilized. Okay, and let's think about the kind of changes that happen to the egg once it's fertilized. So the first thing is exactly what Atal had asked, which is what happens to the zona pellucida. So what happens is the release of these cortical granules that contain, here it says contain enzymes, but it basically consists of ECM components, extracellular matrix components. As soon as the sperm fuses, there's a change in membrane potential, which causes the release of, which causes the fusion of these cortical granules, depositing hyaline and these kind of polymerizing enzymes outside the oocyte, kind of causing this kind of thick barrier to be set up. Okay, so this I have this note somewhere. Yeah, so this fertilization event does involve kind of this change in membrane potential as well. And so we're going from minus 70 millivolts to plus 20 millivolts, which is one way that sperm is kind of, or other sperm start bouncing off the egg membrane. The other way is, of course, this massive now thick layer of the zona pellucida, the vitelline envelope, which also acts as a barrier against more sperm. Okay. So this is what you're looking at here is a sea urchin embryo, and below we're looking at a sensor. as soon as fertilization happens, you can see this sensor light up, yeah? And this sensor is recognizing calcium ions. And so I'll play this again. You can kind of see, as soon as there's a influx of calcium, you can see the vitelline membrane starting to expand. Yeah.
So I have a question here.
Yeah.
Does the vitelline membrane have any receptors on its surface?
So the vitelline envelope, I should call it. It's not a membrane.
Sorry, yeah.
Yeah. It's not
envelope.
Yeah. So of course the oocyte itself, the oocyte membrane has receptors. It has many receptors, and we'll go into that very shortly. The vitelline envelope has no real high affinity receptors. I mean, you can call hyaline a low affinity receptor for various factors, but yeah. So not really.
Okay. So, and that's not different between species. It's more or less conserved.
Yeah, it's more or less conserved. Yeah.
Okay.
Yeah. The shapes of different oocytes are very different. This vitelline membrane, sometimes, for example, in Drosophila is very hard, and it will lift off the newly fertilized oocyte. But this kind of process is fairly well conserved and fairly much dependent on calcium. Okay, and so these calcium influxes are very, very important. I should say that cortical granules contain mucopolysaccharides, and that's and so once it's put out and exposed to the external environment, these kind of gel to form this kind of envelope here. So, as I mentioned, fertilization, the first event that happens at fertilization is this lifting of the vitelline envelope, but also metaphase 2. This metaphase 2 arrest is released. Okay, and so in us, in mammals, what we're looking at is where all of these events are taking place as we go to the uterus. Fertilization happens, of course, in the fallopian tubes, and which is very close to the ovary. What happens on fertilization is, as I mentioned, metaphase 2 is this meta-2 arrest is released, and as I mentioned earlier, the oocyte progenitor doesn't give rise to 4 oocytes. It only gives rise to 1 oocyte. During meiosis, as you know, the cell will give rise to 4 progeny. These progeny are essentially polar bodies, yeah. And so each cell division, each reductive division is one copy of the genome is being extruded into this tiny cell here, which is called the polar body. When we reach metaphase 2 arrest, this last cleavage, this last separation of the chromatids hasn't happened. Fertilization first enables this sister chromatid to separate, and so you get another polar body extruded, and then the maternal pronucleus with a n number of chromosomes is then deposited and allowed to fertilize with the paternal genome. Let me know if you have any questions on this. Yogesh. Okay.
Yep. I have a question related to just previous, like polyspermy. there are two different types of the polyspermy, like slow block and fast block. Like one is changing electrical potential near the outer membrane, and another is changing the shape. So in which scenario or whose side decides which one to do, like out of two?
It does both.
It does both.
It does both.
Okay, okay.
So,
So the changing outer membrane, that's the first, and next is the changing the membrane potential. That's the second step.
It probably is the other way around. Yeah, I feel like membrane potential can be changed much faster than kind of the deposition of mucopolysaccharides. But so I think they're all part of the same process. Yeah, the change in membrane potential happens because of this kind of change in calcium concentration and change in kind of receptor activity, which is necessary, I think, for the fusion of the cortical granules to the egg membrane. Okay.
Okay. Okay. Thank you.
Okay.
Raj, I had a question.
Yeah.
What I would say directs the movement of the sperm? I mean, through the cervix and the uterus, and into which of the fallopian tubes to go to? Because, in subsequent months, either of the one ovaries release the egg.
Yeah.
How does the sperm know where to go? I mean, what directs the movement?
Yeah, I don't know. Sorry, Anjum, I don't know. Maybe, I mean, of course, there's not just one sperm. There's lots of them, and so you can imagine that, some are going the other way, some are getting lost, some are being killed, and some are just some actually make it to the egg. Yeah, and so probably just that sheer number is enough so that it doesn't really have to make a choice. Yeah, there is a chemotactic activity as well, but I'm not sure how when that comes into play. Yeah, I have a feeling it's quite short distance.
Thank you.
Okay, Meghna.
In that image of the sea urchin sperm versus the mouse sperm, only the sea urchin has actin in its sperm, which forms the acrosomal process, which we don't see in the mouse.
Yeah.
What exact purpose does that serve, and why the absence of it in mouse does not have any effect on the outcome?
I don't know. Yeah, and my only answer would be conjecture. Yeah, and I think you could conjecture as well. So why would you think? What's different about sea urchin and a mouse?
I really have no idea.
So anyone else want to chime in? Meghna.
Would it be
to do with external fertilization, internal fertilization?
Could be something like that. Yeah. Yeah. So I think it's something like that. But then, I mean, the experiment we'd have to do is look for acrosomal processes in animals which are externally fertilized. And so I for example, I don't know what a fish sperm structure is like, where you do get external fertilization. Yeah, I don't know, Meghna. That's a good question.
Hi, Raj.
Raj, the shape of the sperm heads is also different for both the species.
Yeah.
And I don't know, but there might be a factor of directionality coming in here, because the mouse sperm has a pointed tip which sort of directs the angle of fertilization, where such a thing is not there in sea urchin. So I don't know why that is required, but this might play a role in something similar.
Could be. And the way to kind of start answering those questions would be just to look at other sperms. So look at a frog or a fish, lamprey, for example, and try and find the evolutional sperm shape. Nishan, you had a question as well.
Yeah. Coming back to the thing of how the sperm finds the egg. Do these sperm like exhibit like run and tumble like motion, or they're purely persistent motion?
It's fairly persistent motion. I mean, they're forced to go in one direction basically by the tail.
But they can't like change their direction at any point, like the way back.
No, I don't think so. I don't think they can.
Okay. It makes sense also because the tail is only like it's oriented only one way, whereas in bacteria the flagella can kind of go all around the cell.
Yeah. I mean, you'd have to have kind of a rudder here, I guess.
Yeah.
To be able to direct the tail motion in different directions.
Okay, thank you.
Okay, where was I? 1 minute. I'm just gonna. Okay, so where were we? So this is a. Last week, Nivedita asked me this question, what these two cells are. And I said they were follicle cells. They're not. These are, of course, polar body cells. Okay, and these have been extruded by the oocyte once metaphase arrest is released. Okay, so let's think about this release in metaphase 2. So it's known that calcium releases this meta-2 arrest. And calcium, how is calcium triggered? So one thing the sperm brings in is this phospholipase C zeta that's in the sperm head. This causes the release of kind of IP3 from PIP2, that's on the egg membrane. This IP3 then triggers the release of calcium, store-operated calcium, which sets in motion all of the subsequent steps. Okay. Okay. So what else does it do? So what else does calcium do? So let's think about what changes need to happen to the oocyte to make it a zygote, okay? So the first thing that needs to happen is the parental genomes need to be reprogrammed, okay? The reprogramming, and so they need to become totipotent. Can someone tell me what totipotency is? And how that differs from pluripotency and multipotency?
A totipotent cell can change into any cell. Like zygote essentially is a totipotent cell. Pluripotent cell, they give rise to a specific line of cells. For example, if I'm not wrong, a hematopoietic stem cell is a pluripotent cell because it can give rise to less than totipotent but more than multipotent cells.
Okay.
And multipotent cells, I guess, can give rise to 1 or 2 cell lines. I'm not sure about multipotent.
All right. So totipotency, a totipotent cell, you're right, can give rise to a whole embryo. And a whole embryo doesn't necessarily need to be the kind of the somatic structures that you see. It also includes the gametes as well. And in humans, or sorry, mammals, or anything that's internally fertilized and develops internally, it's also the placenta as well. Okay. Pluripotent cells can give rise to the whole embryo. Okay, and so a pluripotent cell doesn't necessarily give rise to the placenta. okay? But the pluripotent cell can give rise to gametes. A multipotent cell is, as you said, it has a slightly more restricted potential. Multipotent cells can be, as you said, hematopoietic stem cells, could be neural stem cells, and they'll give rise to a few different cell types. And then below that, you have bipotential cells, and then just a unipotential cell, okay? So the parental genomes need to be reprogrammed for totipotency. And so remember, the paternal genome is packed by protamines, and is heavily methylated. So upon fertilization, it needs to be almost completely demethylated. What is left are methylation marks at imprinted loci. These need to be maintained. So have you all been taught about imprinting, genome imprinting? Okay.
I'm not very sure about that, so I would like to
Okay.
In the context of X chromosome inactivation.
Yeah. So it's something like that. Yeah. I think when you have molecular biology classes, I'm hoping you'll get introduced to genome imprinting a little bit more. But you're right. You know, X chromosome inactivation is kind of the extreme example. But you also get some loci where more than one copy of that loci is detrimental to development. Okay, and so one of these loci, one of the copies need to be inactivated, and that's imprinting. So this is one thing that needs to happen. The paternal genome needs to be demethylated and repackaged with histones. Also the cell cycle machinery has to switch, and so we've been undergoing meiosis up until now, and so this has been a reductional chromosome segregation where you're going from 2N to N, and now you want to switch to what's called an equational segregation, which is mitotic segregation. And so what we're doing here are these steps here. The calcium is important for kind of basically exiting the metaphase 2 for the maternal chromosomes. The maternal chromosomes are protected from demethylation by this protein here. The paternal chromosome, paternal pronucleus is hypercondensed. Soon after it enters, you have exchange of protamines for histones, and you have active demethylation by this protein here, TET3. After this, you're getting passive demethylation and DNA replication as the zygote cleaves. Okay. How is this switch from this kind of meiotic mode to mitotic mode taking place? And what I'm gonna do is just introduce the names, okay? If you're interested in this, and it is very fascinating, I kind of recommend you read the papers or read the review that I've cited here. So meiosis seems to depend on this complex between cohesin and REC8, which wraps around the chromatin. Upon fertilization, separase breaks the cohesin bond, destroys REC8, and REC8 is then substituted by this other protein, SCC1. SCC1, again, complexes of cohesin to wrap around various chromatin, but somehow triggers now this kind of non-reductive, this mitotic mode of segregation for the chromosomes. Okay. So as you can tell, I'm not an expert on this, and so I will. This review is already up in the Moodle page. I'll put some more reviews up for you if you're interested in this behavior. The next thing that has to happen is formation of a centrosome. So in almost all species, the centrosome is absent from oocyte meiotic spindles. And so I think all of you know what a mitotic and meiotic spindle is, yeah? Anyone doesn't? You can use the thumbs up icon or the tick icon or a cross. Thanks. Okay, thanks, Grish. So cell division is coordinated basically by spindle. And a spindle is a centrosome that nucleates microtubules which hook on to the different chromatids. The chromatids will divide. Sorry, I kind of lost where I was. And so the chromatids will be pulled apart by this spindle that's organized. Okay. So in mitosis, the spindle is organized by centrosomes. Centrosomes contain centrioles. It just so happens that oocytes seem to have lost their centrioles. They have an acentriolar centrosome. And so you can see here this much more bushier spindle that is completely acentriolar. Somehow, so fertilization brings back the centriole. In mice, if you remember the structure of the sperm that I showed you, the centriole is attached to the paternal genome. In mice, that centriole is completely destroyed upon fertilization. So here in mice, the centrioles are generated from scratch. But in humans, this might not be the case. So you get some centriole that is brought in with the sperm, and it could be acting as a template for subsequent centrioles. And so early in development, so after fertilization, the division is fairly acentriolar, but as development proceeds, you start getting the centrioles back. Okay, so you get centriolar centrosomes come back. The other thing that needs to happen is reinitiating gene expression. The maternal transcription stops at the end of oocyte growth before maturation, but restarts after fertilization. So what oogenesis does is to bring in maternal RNAs, proteins, to carry through the egg after fertilization. These products may only be needed in the mouse 2 cells, so 1 cleavage, in humans for 4 cells, so 2 cleavages. But in the xenopus and in fish, it's needed for a long time, for about 8 hours, once the blastocyst stage forms. How is this working? So the other thing that happens is once zygotic transcription starts, the maternal RNA must be destroyed as well. And so it seems that all maternal RNAs that are stored in the oocyte have particular sequences in the mRNAs. These are called cytoplasmic polyadenylation elements. And these act as control elements for particular proteins that are activated upon fertilization. which then will either activate, so that will then destroy maternal RNAs. At the same time, there's a particular chromatin remodeling event that's happening that uses this SWI/SNP complex, and this opens up the now zygotic genome for transcriptional activation. Okay. Any questions so far?
Raj, so
yes.
maternal transcription is stopping at the end of oocyte port. So
yeah.
From that point onwards till maturation, is it mainly nurtured by maternal factors from the maternal cells?
Yeah, yeah. Also, it's remember for a lot of for most of the oocyte's life, it's wrapped around by kind of support cells. For example, in humans, these are follicle cells, which and cumulus cells, which will give nutrition as well to the oocyte as well. Okay, but you're right.
So if
yeah, go on.
So if that is the case, so I was wondering why the maternal RNAs must be destroyed after fertilization and not just after the stop of oocyte transcription.
Yeah, so I think some of those maternal RNAs are important for development. Yeah, and so, for example, the example of Xenopus development, you have cortical rotation, which brings in two sets of RNAs which are localized within the egg, and so localized RNAs in eggs do a lot, and I think that's probably why many maternal RNAs need to stay around for a longer time. This is
okay. I was
yeah.
No, I was actually, I think I'm a bit confused about the term maternal RNAs. Does it involve the RNAs which have been provided by the support cells, or also the ones which have been generated within the oocyte before the transcriptional arrest, or is it both?
It's both. It's both. Yeah, yeah. So it's any RNA that's there in the egg prior to fertilization.
Okay.
Okay. So
hello. Sorry.
Yeah.
Yeah, I had a doubt. So how are these maternal RNAs destroyed? And so if you're talking about it in the egg stage, so how are they differentiated from the, say, for example, paternal mRNAs? I mean, yeah, RNAs.
So there's almost no paternal RNAs. So the only thing the sperm provides is a pronucleus and perhaps a centriole if you're lucky. Up until a year or two ago, it was thought that the sperm didn't even provide mitochondria as well, even though the sperm head is packed with mitochondria. So it's not really providing any RNA. So all RNA is coming from the mother at this point.
Okay, so when does the paternal RNA start getting transcribed, or at what stage would that happen?
So when it's no longer paternal, when it's now the embryonic.
Okay.
Yeah.
So when it's one, that's
yeah. So when the two pronuclei are fused, okay.
Okay.
And in the mouse, that 2-cell stage, some of the RNA that's transcribed has come from the father. Some have come from the mother. Some RNAs come from both. But by this time, it doesn't really matter,
I think
yeah.
Yes, the formal term for this stage is maternal to zygotic transformation.
Yeah, exactly. Yeah. So it's called a few different things. It's called zygotic gene activation most of the time, ZGA. Some people also use, some Xenopus people and fish people use the phrase mid-blastular transition because in those animals, the activation of zygotic genes happen during the mid-blastular stage. But you're right. It's all of them are maternal to zygotic transition.
Raj, I had a question.
Yeah, Tal.
So when this maternal degradation, maternal RNA degradation is happening, how is the cell able to differentiate if it is destroying the maternal RNA only or the embryonic RNA? Should there be a marker which decides that?
Yeah, so I think it's the use of these cytoplasmic polyadenylation elements. The kind of protein machinery for that is restricted as well. And so it's one of the things that are provided as the oocyte grows. These two genes, MIS2 and CPEB and DAZL, yeah, and on fertilization or once zygotic genes need to be activated, these are phosphorylated or they're dephosphorylated to cause the destruction of the maternal RNA. Okay.
Yeah.
I have the follow-up question. So when you talk about when we talk about the zygotic gene activation at the 2-cell stage or the 4-cell stage in mice or humans, so after this the next division when the whole sort of these cell cycle stages will be happening. So at that time, the whole chromosomes and all sort of things will condense. And how does the cell remember which genes need to be activated again in the next stage, in the next division? Because these genes would be important in the next division again. So is there any sort of like bookmarking or sort of things?
Yeah, I think there's always kind of marks. There's methylation acetylation marks, which kind of mark out genes that are being used or not being used. This is so it's a good question. And as you can see, I can't really answer it so clearly because it will take a little bit longer to answer. But you're right. There are these marks that are on regions that need to be activated, which allow the cell to remember what it was doing at that particular point. Okay. Is that okay?
I did have a follow-up question.
Yeah, go on.
Yeah, so you had mentioned that there was a demethylation happening before this stage.
Yeah.
So now I'm wondering, so how did those methylated sites still remain for gene activation?
Yeah. So some of these are good questions, and you're gonna start seeing me flounder as I can't really answer these so clearly. So most of the paternal genome is demethylated, but things that need to remain methylated somehow escape this demethylation. And so these are genes where copy number of the gene is important. So they're imprinted loci. Of course, as soon as you have the zygotic genome, some of these then become methylated again or passively demethylated during replication. And this is kind of done through the regular methylation, demethylation machinery here. But this kind of a massive demethylation that happens in the paternal genome early on upon fertilization is mediated by TET3. But there are a few regions of the paternal genome which somehow are protected from demethylation. And these are the genes that need to be imprinted.
Okay.
Yeah.
So is that what we call epigenetic memory?
So it's kind of epigenetic memory, yeah. Here it's a little bit easier because it doesn't have to remember too long. Okay, it just needs to remember from one division to the next. But yeah, it's kind of that.
Okay, thank you.
Sorry, I just see it now. I suddenly have remembered that those imprinted loci, their methylation is maintained by these particular proteins here, okay, ZPF57 and TRIM28. All of these, so both TET3, ZPF57 and TRIM28, when they're knocked out in mice, cause a complete arrest in development, okay, and so they're absolutely vital for development.
Okay, thank you.
So that, and so the other thing that now needs to happen is that we're going from asymmetric to symmetric cell division. This I'm going to skip through quite quickly. I think those of you who are interested in actin-myosin meshworks and how they interact with vesicles to initiate kind of spindle asymmetric cell division in the oocyte should probably have a read of this review here, because it is very fascinating, and I don't think I can do it justice so quickly. Okay, but essentially these vesicles act to nucleate actin, and which causes the position to be, the spindle position to be very eccentrically localized in the oocyte. And so remember, you need this very eccentric localization of the spindle, because soon after metaphase 2 is lifted, this metaphase 2 arrest is lifted, the polar body needs to be extruded, okay? And so this complex needs to be very close to the surface for that extrusion to take place. Yeah. Soon after this happens, we go back to normal business, basically, where now the migration of the chromatids happens because of microtubules. The male pronucleus is associated with a centrosome, and the centrosome nucleates a microtubular aster. The aster is this kind of star shape of microtubules that emanate from the male pronucleus, and these asters are used as tracks to bring the maternal pronucleus closer to the male pronucleus, so that then the chromosomes can fuse. Okay. Any questions here? Good. So this is kind of the fertilization process. What happens next is cleavage. And so here I'm talking about the need from going from asymmetric to symmetric cell division. But we need to go back to asymmetric cell division once we start cleaving the embryo. So I'm going to talk very briefly about cell cleavage now. And just show kind of what we're thinking about here, okay? We're thinking about cleavage, separating the cells so that we have now many cells with which to start development, okay? Here the sea urchin has a very regular cleavage. This is from the National Institute for Basic Biology in Okazaki in Japan. And you can see here Xenopus cleavage as well. So it's kind of regular, but then becomes deregularized at some point, as we've talked about earlier. So the type of cleavage you get depends very much on yolk, okay? And so of course some embryos don't use yolk, and we'll come back to those. But essentially you have two types of cleavage, holoblastic and miroblastic. Holoblastic is usually when there's either no yolk or the yolk is very evenly distributed. And there, what this refers to is that the cleavage is complete. So when cells divide, they divide completely. Where you have either very dense yolk throughout most of the cell, or the yolk is deposited in different places, you get what's called mirroblastic cleavage. Yeah, and here cell division is incomplete. So for a long time, the embryonic cells still will retain some connection to the yolky cell underneath. Okay, so even in these, here you can subdivide holoblastic cleavage into either, depending on where the yolk is, into isolecithal. So that can be radial, spiral, bilateral, rotational, mesolectical, which is radial, and then telolectical, here when there's dense yolk. And this is usually associated with mirroblastic cleavage. So I've taken this paragraph from Gilbert's developmental biology textbook, and he writes that in 1923, the embryologist E.B. Wilson said how little we know about cleavage. To our limited intelligence, it would seem a simple task to divide a nucleus into equal parts. The cell manifestly entertains a very different opinion. So as shown here, different organisms cleave in different ways. It depends on where the yolk is. And there's factors, of course, in the egg cytoplasm that affect the influence of that affect the angle of the mitotic spindle, for example, and where cleavage planes happen. So what all of this is saying is that generally we don't really have a good idea what's specifying, what's kind of directing the cleavage planes. So we know that to get particular kind of patterns of cell division, you need to orient the spindles. So these are the cleavage plane, the chromatids as they divide. You need to orient them in particular ways. So cleavage patterns, and so I started writing this, and I realized that this was kind of very, this was never ending, okay? Cleavage patterns depend on cleavage planes, which depend on spindle orientation, which depend on ... And so there are unknowns there on what is determining the cleavage pattern of an embryo, okay? So I think that's that. We've come to the end of this class. If you have any questions, you can ask them now.
I had a question regarding the paternal genome, paternal pronuclear demethylation and maternal pronuclear then being protected by it, but then again, like when they fuse, it undergoes a kind of transformation. So I got a little confused, like why is the maternal genome then being protected and then only to get transformed post fusion of the two?
Yeah, that's a good question, and at the moment I can't answer that. I have to say I was kind of confused by it as well. And so I'm wondering if you need particular marks on the maternal genome to be retained. But I think I need to talk to Dimple a little bit more about this to fully understand it.
Okay.
Yeah. Okay. Any other questions? Okay, great. So if there's no questions, then we'll call it a day. If you can think of any questions, then please write to me. I've started putting up these lectures on YouTube. The link is on the individual lecture pages. So if you've missed or you suffered from bad connectivity at some point, you can catch up the lectures just on the Moodle page, okay? So with that, I'll see. Bhavnesh, you just unmuted.
Yes, sir. Sir, I have a question. After fertilization, maternal mRNA has to be destroyed.
Yeah.
For maternal to embryonic transition.
Yeah.
But if somehow this maternal mRNA get destroyed before fertilization, so the oocyte will start transition towards the embryonic stage.
So it will undergo some of the, it'll try and do what it can, okay? But remember, some of that maternal mRNA is used for localizing particular patterning cues for the embryo later on. And so it may be difficult to do that experiment. What I should do is look at, the experiment has been done. So the proteins that I mentioned bind to the cytoplasmic polyadenylation element have been mutated, and they and so it's the opposite experiment, just prolonging maternal mRNA, and that also arrests development as well. Okay, I don't know what happens when you remove maternal mRNA slightly before fertilization. I mean, if you removed it very early, then of course I don't think you would get you'd get development happening at all. But yeah, I don't know.
Yes, sir.
Yeah.
Sir, but if somehow we have a oocyte and we destroy the maternal mRNA, and we also add whatever material present in the sperm had except the genetic material, will then the transition will take place up to some extent?
So you can activate the egg by different means, okay? And so it depends on what transition you're talking about. You can, of course, you can activate eggs artificially so that you release the metaphase to arrest. You can allow some level of development as well. So, of course, there are a number of species which develop as haploid embryos. So, soldier ants, for example, certain wasps. Yeah. So yeah, there are ways that development can take place. I'm not sure it can happen maternally, though. Yeah. What I need to investigate is what happens in parthenogenesis. And so there has been reports of fusion of two eggs to make an embryo in mice. And what I'll do is kind of investigate how that was done and report back next week, okay?
Okay, sir.
Yeah?
Yes.
Okay. Okay. Well, if there's no questions, so thank you very much, and I'll catch you on Monday, okay? Have a good weekend. See you.
Thank you.
Thanks.